Let's gather around this table for a moment, and then we're underway. Very simply put, these courses they add up to a once-in-a-lifetime thing for me, even though I've been teaching nature, and I taught mathematics before that, and some physics and other things. Since the late 60s, we picked grasses, sedges, woody plants, and mosses as groups that I thought were both foundational in every habitat and known, but perhaps not as well known as they should be. There are a surprising number of subtleties in woody plants, and woody plants have a sort of unique challenge a unique offering in that they are present every day of the year and in a forested landscape in every habitat you go into. So you can always find them and you can't get away from them, but you need to know buds and twigs. Bark doesn't cut it for me. Bark is useful. It's kind of nice. It's there. The trees work hard to make it stay on and so on, but it's not that useful. But buds and twigs, leaves, flowers, and fruits, and even more, very telling to a statistician, you're always working with very incomplete information. At any time of the year, you only have a part of the whole presentation of the plant. And sometimes it's enough and sometimes it's not. We want to start with vocabulary because I think that's where you always start. So what I'll have you do for this 15 minutes, work individually, unless you really want to work with somebody else. Take one plant off the table, and you'll know its name. Make two quick sketches, one how the leaves are arranged on the twig, and the other the shape of the leaves, and then describe it, label those sketches. If you want words for leaves, in our woody plant book over there, the first couple of pages are a visual glossary, and there's a visual glossary of leaf words and arrangements that you can go right to. And then in 15 minutes, we'll regather here, you with your plants, and we'll just go around the room. The object for me in teaching is always to get people to talk plant first. Looking is wonderful, and we'll look a lot, but for the way we understand, we have to develop looking and talking about looking simultaneously, or it doesn't work. Ladies and gentlemen, a session for talking leaves, and we'll just go around the table. So tell us about your plant, and tell us some of the words you use to describe it.
So I chose the red osier dogwood.
And smaller teeth in between, and very petioles.
Number one, the birches are closely related, and sometimes when we're not looking, they get even closer than that. So the birches are a genetic complex of things with chromosome doubling. And that basically means that something with 4 sets of chromosomes, or white birch with 6 sets of chromosomes, but other numbers too, are made from pieces of other species. Dr. Frankenstein did that in his laboratory, but he was making monsters. Nature does it all the time, and we simply call them plant species. And the birches and quite a number of other groups are made from the same elements, so that when we look at a yellow birch and a black birch, we say, well, they're awful hard to tell apart, though the adult bark is different. And then we say, well, yellow birch is a tetraploid, 4 sets of chromosomes. It's probably got a whole black birch genome. sort of available, tucked inside, that it can access. And there's a reason these things are close together. And that's, that's how evolution works, and it's kind of fun. As the willows do much the same thing. Plants have a very simple, I would say very repetitive, rhythmic architecture. They're made from very few components and endless variation on those components. Once you have talked stem, leaf, root, and a few sexual cells, you've talked the whole being of a land plant. And just a thing about words, the important thing is to be able to talk about these things. The specialist words like stipule are very useful because they get a lot of mileage quick, and so I think I emphasize knowing them, but sometimes they're not clear, and we can always paraphrase as long as we speak good, clear English. So your accessory leaf-like things near the base is a perfect paraphrase. It tells us all just what you were looking at. And a lot of plants use resin, and they use it as a fungicide, an insecticide, a preservative from wood, and they also use it as a drying, but speak English to the nice people. They use it as a drying resin, like shellac, which comes from bugs, that will harden, so that they can cover their buds with it in the conifers, and then it forms a hard, waterproof, protective, bug-proof surface. But it often has to be liquid inside, so pretty much all our conifers have resin canals, both in the stems and in the leaves. Those pines have a couple of resin canals in each one of those needles running the length of the needles. We don't see, in the deciduous trees, we see resin on the surfaces of the buds as hard resin all the time. Think bayberry, huckleberry, things like that. But the cottonwood and the balsam poplar, the buds, you cut them open and you get a little sticky pool, and it's sticky indeed. Imagine some poor sucking aphid trying to get juice out of one of those buds. It would be doomed. So the pollen grain, once it lands on the style, and it never moves, but it does what plants are really good at. It grows a root, a tube, that has to penetrate down, sometimes quite a long way, and it has to go down and then reach one of the ovules and fertilize. And just to make it tough, it has to fertilize twice. It fertilizes the egg, which makes the embryo, and it fertilizes two nuclei that happen to be hanging around waiting for action to make a triploid food tissue, which is the endosperm. I am putting this all in the new book. My editor says we won't be able to sell in Texas, but but she says we don't sell anything in Texas anyway, so do it.
No science.
That's how all pollen works? It actually sends down?
Pollen is a broader term, so that pollen also applies to conifers and gymnosperms and tropical plants like the cycads. All pollen lands on, and in the gymnosperms, it lands almost on the ovule itself. It actually lands on a drop of fluid and gets sucked into a hole in the ovule as the fluid evaporates. In the angiosperms, all these things except the pine are angiosperms. It lands on a style, and then it grows a tube down. It reaches the ovule. In the gymnosperms, poor old primitive things, they only dominated the world's vegetation for 150 million years, and they've been here for 200 million years, and they still dominate the whole north and the south of the world. But anyway, those poor primitive things only fertilize once. They fertilize just an embryo, and they don't have, they make seed, they store starches in the seeds in other ways, but they don't make this high nutrient endosperm nutritive tissue that the angiosperms make. But otherwise, the story is pretty much the same. Gymnosperms have spanned an enormous length of Earth's history. So if I have time this afternoon, I may talk about this a little more. But when seeds are first invented, they're by things that look like ferns. And in that case, to invent a seed, you have to invent pollen too. The two inventions like wheel and motor go, or wheel and horse go side by side. So all pollen grows a tube down that reaches in or near the ovule. And in the oldest gymnosperms and ginkgo still and some of the cycads in the tropics, nuclei don't just sort of crawl out of the tube, they actually swim out with beating things like that, and they have all the architecture of plant sperms, so there's no reason not to call them sperm. In the gymnosperms and in the angiosperms, the nuclei are just in the tube, and somehow that I don't understand, they make their way to the wall of the ovule and, like a virus, inject the male genetic material into the right cell at that point. So the sperm are old gymnosperms. We would find them in ferns, which are not seed plants, but we would find them in every fern and club moss out here too. Number one, not only does a twig, which I call a shoot, which is a combination of a stem and leaves and maybe flowers, come out of a bud is a shoot, all those bud scales and everything else. So every bud is a shoot. Number two, the leaves within that bud have anatomically, microscopically, buds for the following year in them. So there's at least the anticipation of 1 year, and some people have said that at least in some plants you can find buds for the year after that. So you can find buds within buds within buds. Plants do extraordinarily well at reiteration and at the nesting, at the nesting of form within form within form. This is wonderful. This is the warm-up for a lot of field work, but you're talking woody stuff really fluently. The next thing you will do is to work with one or two other people, go out and find a variety of leaves, not a whole lot, but let's say 5 leaves that are either ones that you've seen here or ones that you haven't seen, but that are as maximally different from one another as you could find. Then what you do is, number one, see if they're on one of the labeled things on this table. If so, that gives you a name. Otherwise, you have to work it out, and we'll work with you. You can start by guessing and hunting, which is an old, powerful technique, and I'm not being ironic. It works really well in natural history. It's one that the pros use all the time. And then what I want you to do is make a sketch of each one of them on a bigger piece of paper and just make arrows or so on, comparing them to one another, the things that one leaf has that no other leaf has, the 2 leaves that might be closer but differ in this respect, and so on. So what we're doing is leaf-to-leaf comparison, and then look them up in any of the field guides, mine or the others, see some of the diagnostic characters, and the ones that seem to work for your leaves, write those on your piece of paper. So it will be as if you were doing a little guide to 5 leaves that you had brought down, and again, work with someone else. Let me say what we're leading to in this. I think of botany as, I think of the working process of botany as you are out there and you see something you're interested in. And often the first thing that goes on, is that you notice the habitat it's in, how it's growing, the color. You don't see many details yet. You may be 30 feet away. But already, because of the habitat and because of the growth form, you have rejected many, many things. Rejection is often easier than confirmation. Knowing something is not an oak tree is really easy. Knowing which willow it is gets much harder. So you've rejected things, and then you come up on it, and that's a second stage. And as you get closer, if you've been doing this for a while, whether it be a bird or a willow, you have a guess as to what it can be, a conjecture. And a botanist without guesses is a poor, poor creature indeed, or is someone just getting started. So, the next step is to guess at an identification, and then you need sooner or later to confirm. Your guess may be right or wrong, it doesn't matter, but the guess is a starting point. In the quantitative stuff I did, the way you studied problems was a cycle. You gathered data, you made a model that suggested what that data might say, you went out and gathered more data, you updated the model, you worked from prior to posterior to better posterior, and you kept bringing in data that improved the model. And the great thing about that is that you're never done, because someone else will find new data and then you've got to go farther. But anyway, one needs to confirm, and there's often a group of characters. So that elm there is not strongly rough on the tips. And looking with a hand lens at what was left of the bud scales and the young branches, one would see white hairs, but one would see no metallic red hairs on it. And at that point, a very good guess would be that it is an American elm rather than a red elm, which is slippery elm. How do you confirm that? The leaves, unfortunately, won't do it. The buds would do it. The twigs or the bark or the fruits would do it. So if you're out there in the field at that stage, you look around to see if you can get some fruits. If it's a mature tree, you break the bark and look to see if it's a chocolate cake with white layers, which is common elm, American elm. If you have no other recourse, you chew the twigs, and if they don't foam in your mouth, it's an American elm. But you have, in other words, diagnostic characters. And then, those usually a conjecture, a guess, and then some diagnostic characters are enough. But we have plants that look very alike one another. The cherries often do, the willows often do, the hickories often do. And at that point, the real gold standard is to have several or as many collections as you can make in front of you and start comparing side by side, because it's eventually side by side comparison that allows you to see the differences and write them down and then internalize them. This is the kind of exercise that could go on forever, but number one, it can last the rest of your lifetime. Putting things side by side is such a fascinating business. Number two, one wants to balance indoors and outdoors and indoors and outdoors. They complement one another so well. When I was helping a friend years ago work out a lot of the flora in some of the more remote parts of the Smoky Mountains National Park, we didn't have that much time, but we found if we balanced a day of outdoor work with half a day or three-quarter or sometimes even a whole day of indoor work, we learned far more the next time we were outdoors again than if we just said, we'll do 5 days of work and then try to figure out what we'd seen. Well, by day three, it was just all swimming in our heads. So this consolidation, solidification, note-taking, in these kind of discriminations, it's not what the books say, and what the books say may well have worked for the person that wrote the book. It's what works for you in your landscape. And that's a long process of getting to know, refining, in and out, back and forth. You've learned too much in the last hour for you to tell us all, because we have one more kind of thing to work on, flowers, this afternoon. But let's go around and just tell us some of the things that you looked at, what you saw, anything you want us to know about them. The plants are just what they are. They're individuals, they're related to other plants. A species, all of these names we use are things we make. And they are useful things if they predict things. In other words, a species is really useful, like poison ivy, if it predicts what happens when you walk barefooted onto it, or something like that. Finding the things that run together reliably enough that they can tell us useful things about ecology, about how we're going to use, whether we should make a pie out of the berries, other features we're going to see, whether the wood dries well, et cetera, et cetera. But that it is really a question of which characters hang together well enough to make predictions. And end of Sunday sermon. You don't really see the twigs there. So the needle has the green part of the needle and then a little stalk, and then it has a pale part of the needle that actually runs down the twig, but is still needle tissue, and sheathes the twig. And those are the needle bases. So those twigs have a sheath of bases running down from each needle. These are all details, but I get lost in them, but I mean, growth is not just a detail. Growth is a miracle, and the unfolding of individuality is a miracle. So these things are deep to me. One of the strategies in deep shade that, say, hornbeams use is to pack their leaves very closely together so they form just a sheet of leaves, and they have little leaves falling into the openings between big ones. Another one which hobblebush uses, and some of the creepers and so on, remember that the light in the understory, there's partially a diffuse light, and plants want to get that, but the real jackpot are those sunflecks moving, like brilliant little fish across the floor of the forest, and they have like 80 or 100 times more light than the diffuse canopy light. So if you're trying to trap brilliant little fish, sunflecks, you may want to set your traps spaced fairly far apart rather than all in one space. And that has to do with the statistics of opening in the canopy, and that's the point at which I got lost years ago and haven't gotten back to it. But this stretch out, spend some money on internodes, but cover a lot of ground with small horizontal light trapping units is a very definite strategy used by unrelated plants. So, ladies and gentlemen, this is going to be a visual lesson on flowers. Number one, all the plants, whether they be conifers, which are gymnosperms, or apple trees, which are angiosperms, they all make seeds, and they all use pollen and ovules. An ovule is just an egg with packaging, with a couple of layers of packaging, to make seeds. The seed was a great invention. End of Devonian, beginning of the Carboniferous, sometime there. Of the existing plants, the only ones that still exist from those early days of seeds are the gymnosperms. The conifers weren't around then, but ginkgo most certainly, and cycads and things like that were. But that was a big breakthrough because you had two dispersal stages. You had the pollen, but of course the pollen has to go to a female flower to pollinize it. But then you had a whole packaged seed. So when you take either ovules or fertilized ovules, which become seeds, they're in an enclosure. And that enclosure is called a what?
Fruit.
It's a fruit for seeds, and in the flower it's called an ovary. A simple term, but a very necessary one, because that's often what you see. What are the functions, what function or functions of petals? Why bother to have petals?
You're the advertising department.
Which means cheaply made and possibly deceiving, but useful.
Yeah. But gorgeous.
Advertising to whom?
Insects. Whatever pollinator.
Yeah. To wind?
No, no need to advertise to the wind.
Human.
Well, that's not really, I mean, they could direct the wind.
The angiosperms have not been enough studied on this. The gymnosperm cones, like pine cones, have been enormously studied. They put them in wind tunnels, they blow smoke around them, they do high-speed photography, they do microanemometry and so on. And they are extremely well conceived, both to create swirls that lift pollen out of the male cones, and then that, like eddies in a river, create little whirlpools that swirl pollen right into the pollination drops of the females. And then later, when the cones are bigger and they open again, there are swirls that lift those winged seeds right out. So they are all wind management devices. You would think that the angiosperms, having co-evolved with insects, would say it's insects forever, but in many different groups of lineages of angiosperms, they re-evolved wind pollination. So the wind pollination in oaks and things like that comes almost certainly as a development from an insect-pollinated ancestor. But nonetheless, the aspens, the oaks, the beech, we put them all together. There's a very considerable list that wind pollination is one of the commonest pollination modes in our woody plants altogether. Sum this up by just going around, and we'll pick up different plants here, and we'll just hold them up, and you individually or all can say anything about them. So for instance, the apple, a big showy flower, the stamens and the pistils stick up above the flower, but it has a really nice refinement. So, when you eat an apple, the stalk is down here. Where are the remnants of the sepals? Are they on the stalk end, or no, they're on the opposite end. So the ovaries actually have been sunk down in a cup, like a rose hip, and they've been surrounded by all this other tissue. Those nasty insects might eat them otherwise. So there's a strong sense among flowers that it was a really smart move to separate the parts that actually need fertilization from the parts where the insects, from the playroom where the insects are allowed to roam in, and where those things like beetles can arrive and just start chewing. The leaf shape and the very inconspicuous teeth are all all black cherry characters. It's a little glossier than black cherry. In botany, if a character is less than 50% reliable, it's junk. But 80% is not bad, that gets you near. Question is, if you have 80% characters, if you get up to much better than 85% or so on. And I'm not sure you do, but again, that leaf shape I trust, the low teeth I trust. This is a compound leaf with how many leaflets?
3? So it's three. So
3 leaflets.
Okay.
In another couple of weeks, the new raspberry canes will come up from the ground all over the place. So these are old canes, they're with flower buds, bless them. But the new canes will come up, and the leaflets will have 5 leaflets almost uniformly on the new canes. So this really has 2 leaves that you wouldn't think belong to the same plant, depending on whether it's on a second-year stem or on a first-year stem. One of these, normally the one that continues long and not have a very long beak. And so both of those, nice, well done. So good silky willow characters.